All right. Yep, I'm Scott. I work at Merck. My team is a bioinformatics team. We sit within Research and Development Sciences IT, within Merck Research Laboratories IT. Our organization is essentially an interface where we provide support and services for research laboratories that do the amazing work in Merck Research and Development. And one set of those teams is in the early discovery space, and their needs are quite unique because they need to have scalable solutions for discovering, like target discovery, compound discovery, what works, what doesn't. And so they've turned to a technique called DRUG-seq. This was published by a team from Novartis back in 2018. And it's essentially horizontal scaling of bulk RNA-seq. Essentially a cost-effective solution for processing hundreds up to thousands of bulk RNA-seq experiments in a single go. And it works by performing your experiments in multi-well plates, attaching a barcode in the reverse transcription step, amplifying sequencing, and at the other end, they generate this FASTQ data that they need processed into a counts matrix of unique molecules per well. And so that's where my team comes in, is to source or develop a solution for processing this data. So just a quick overview of what the data looks like. It's paired end sequencing, but only one read contains the cDNA sequence. The first read contains the barcode and UMI. And the observer among you might recognize this as similar to single cell or spatial transcriptomics. So in theory, we could use 10x cell ranger, except that we're kind of locked into their set of barcodes, which doesn't match the barcodes that we use for the plates themselves. Now, the authors of the DRUG-seq protocol did state that they have their own methods available on reasonable request. We did issue what we believe is a reasonable request. Still waiting to hear back, but in the meantime, we looked into some of the off-the-shelf solutions for similar data, to try and learn what works, what we like, where the gaps are, and if necessary, and it turned out to be necessary, integrate all of our learning into a brand new Nextflow pipeline. So starting off, ST pipeline is a spatial transcriptomics pipeline from 2016. It's a Python-based pipeline that handles exactly this kind of data set. You give it a set of barcodes, you give it the FASTQ, and it'll spit out a kinds matrix, which is exactly what we want. Zoomies is similar. It's a more R-based platform, but it takes the same inputs, spits out an RDS, which you could manipulate to generate the necessary counts matrix. And then Star Solo is actually inside the popular Star RNA-seq aligner. It's a turnkey solution that promises greater speed than 10x cell ranger, and it lets you specify your own barcodes, which makes it perfect for this kind of problem. Now, all of these pipelines have their strengths. ST pipeline in particular has very robust QC reporting, which we as researchers love. And Star Solo is quick to set up and use out of the box. But they all have some drawbacks, which I'll get to in the next section of my talk. But I do want to introduce the Nextflow pipeline DRAGoN, which makes a few key innovations relative to the off-the-shelf solutions. The first is that it does the plate demultiplexing, the well demultiplexing, as an initial step. And this enables two things. One, it enables horizontal scaling in compute to mirror the horizontal scaling in the lab. And then it also lets us observe QC metrics at each step for each well. So if, say, one well has some contamination in it that's not present in the others, you can pick that out, and instead of redoing the entire experiment, you could just remove that replicate from the analysis. So super useful to the researchers. And if you don't like this representation, I've got a metro map for you. All right, so let's look at the benchmarking between all 4 of these solutions, and where they excel and where they kind of lag behind. So for these tests, I used a data set that was published by another team at Novartis that was using DRUG-seq. It's about 500 million reads per well, per plate, 9 plates, 360 wells per plate. So an ideal testing bed for all of these pipelines. And I looked at metrics of runtime, memory usage, and just as a sanity check, how many UMIs were generated at the end. So starting off with runtime, Zumi's, DRAGoN, and ST pipeline performed relatively similarly, but you'll notice that ST pipeline is way off the chart, and this y-axis is in log scale. And when I dove into it using a smaller subsampling of these data, I observed that in the duplicate detection step using the UMIs, that step scales by order of n squared. So as you increase the number of reads, the time it takes to deduplicate goes up by that squared. And when you scale up to the typical output from a NovaSeq, you start to get into the weeks plus processing time. It's good for a small lab, not so good for teams that are running on deadlines. Okay, so ST pipeline is out. But what about Zumi's? That ran pretty fast at a cost. So this is the memory plot. ST pipeline, Star Solo, and DRAGoN are all at 32 gig per process, which makes sense. That's the size of the GRCH38 index. Zumi's is all the way up here. And it turns out that that's also due to the UMI collapse. It uses a Hamming distance matrix, which is a full matrix in memory. And again, at the scale of NovaSeq, you're trying to allocate terabytes of RAM. So that's out. And then sanity check, all of the pipelines are producing the same total UMIs across all the wells, more or less, a little bit of variation here and there. All right. Now with Star Solo, this was run as a standalone, so there's no upfront QC and filtering or adaptive trimming. So that would have to be built in, and that would increase the runtime for Star Solo as well. All right. So quick summary of the benchmarking. I have about a minute left, so I'll just quickly go over and reiterate the main benefit of upfront demultiplexing in DRAGoN, which is that you get these per well QC metrics. So you could see, if you have a low UMI count in these 4 wells, well, yeah, because those are negative controls. That makes sense. Or you've got this weird mosaic pattern of reads coming in because there was a problem with how you programmed the machine that actually loads the plate. So, and there's also like text reports and multi-QC reports summarizing all of this as well. So, in conclusion, DRAGoN's upfront demultiplexing divide and conquer strategy provides horizontal scaling of your computational workflows, and that very important per well QC. And with that, I'd like to acknowledge everyone who's worked on this project from the IT and the compute side, and I'm happy to take questions.
Thank you, Scott. I see someone, Ken, very in-depth question. Who drew the DRAGoN logo?
So that is actually an icon from within PowerPoint.
Okay, I see Francine's typing as well. Do we have any questions from the audience?
Thank you for the good presentation. Because I also use to try to align the DRUG-seq generating the data with the different way. But I find when just do the downstream analyzing, there have some empty jobs is still existing on there. It's because they are single cell level based. So the matrix is not like the regular book RNA-seq generating. It's more like the single cell is more sparse. So is your DRAGoN pipeline can solve this question, or we still need to do to solve like in the single cell level?
So I missed a few words in your question, but essentially the counts matrices that DRAGoN produces are that there's two formats. There's the sparse matrix, similar to what you'd get from 10x, as well as a dense matrix. So you can load them into whatever downstream analysis software you have, even just a basic R script.
Yeah, but the seq2 is not working very well on the sparse matrix.
Yes, so and that's an interesting point that you bring up with DSeq2 is, so one of the features that DRAGoN has is that it's able to generate fractional counts, which DSeq2 also doesn't really like. You'd have to coerce it to integer. Now, you could load in the dense matrix, it's like a TSV file, coerce it to integer and load it into DSeq2. You could also load it into edge-R as is, and use that edge-R lemma workflow.
Thank you.
Do we have any more questions in the audience?
So I just wanted to get your thoughts on like the increasing prevalence of these types of UMI tags for drug compound libraries and combinatorial chemistry. Do you see this as like a major feature in terms of the way like large-scale screening gets done moving forward?
I mean, the UMIs are usually to like correct for biases that might be introduced during the library amplification step. So I do see it as a net benefit. Perhaps we need more robust tooling for handling UMIs. And to the credit of the community, UMI tools is fairly robust at handling these kinds of data sets.
So I've got a question as well. So providing you can get past Illumina's lawyers for the trademark infringement and continue your work, this seems like a really nice use case for GPUs. Have you looked into any of that? Is there any possibilities to get that additional speed up and bring runtimes down?
Yeah, that is an excellent question. So I haven't like fully considered GPUs for this workflow, but this is an excellent use case. And in fact, even just multiprocessing on CPUs can bring some of the custom programs within the DRAGoN pipeline down in runtime.
Awesome. Thank you, Scott.
Thank you.
Cheers.